Hello and welcome to another Odds and Ends episode. In this episode, I'll be talking about the pests of grapes and the conditions that shape their evolution, as well as their management and control. Let's get started. Grapevines, upon exiting dormancy, have various physiological stages that offer cues for timing of arthropod activity, and by virtue of this, offering proper timings for various interventions to manage these arthropod pests. Various insects favor grapes for many different reasons. The tarnished plant bug feeds preferentially on plant materials that are released by different plant organs. For instance, flower buds are coated with pectin, and the tarnished plant bug favors the consumption of pectin. The sap of various cultivars, especially those abundant in sugars and amino acids, are attractive to different bug species. Berry development is uneven as well, both in the wild and domesticated species. This provides a long-term source of sugars and food sources for different arthropod species. Hence why various insects and other arthropods preferentially seek out these species as a common food source. Because of this, there's been over 1,000 scientific papers made on vineyard insects and arachnids from 1972 to 2010, with most of it focusing on arthropod pests. As a consequence, very little is known about the biodiversity of life that exists within a single grapevine. There is, of course, an exception to this rule. In Quebec, a full study of every single insect within a vineyard system. This is because many of these are vectors for the disease called phytoplasma, and it's recorded that there's 60 leafhopper species within Quebec that impact grapevines by feeding on them and by passing on diseases. There's also 124 beetle species, as well as 97 species of spider, 73 weevil species, 20 species of ladybug, and 59 species of leaf beetle. This genetic and species diversity may represent a possibility of a great many natural enemies that can be used as biological controls for various grapevine pests. And it was speculated that this could be used to manage pests with far fewer insecticide uses in low input systems, organic systems, and biodynamic systems. Thanks to the extensive research on what pests affect grapes, it was found that there's 150 arthropod species that are pests of grapes worldwide. Damage caused by arthropod pests fall into two categories: reversible and irreversible. An example of the irreversible would be the glassy-winged sharpshooter. This species drinks the sap of different grape species. The damage itself caused by the insect is not exactly dangerous to the plant, but it does vector the bacteria species Xylia fastidiosa, a causal agent of Pierce's disease. Leafhoppers also do the same thing, vectoring phytoplasmas, a family of bacteria species. Once bacteria get inside the plant tissue, you cannot treat them, and the only management option is to destroy the plants to prevent any more spreading of the disease. For most other species, the damage is reversible. Species that do not vector disease but still feed on the plants do damage related to their population. Berry moths, mealybugs, and aphids are such examples. The most common way to deal with these insects is through broad-spectrum insecticides, but several alternatives have been made as well, and I'll be covering these next. The first new class of chemicals we will discuss will be semiochemicals. These are chemicals that mimic sex pheromones of moths. In vineyards, they're used to detect, and in some cases, disrupt the mating cycle of these insects. The way this method works is that the males get attracted to the stick releasing specific pheromones and try to have sex with it instead of females, because the mating pheromone dispenser releases heavy amounts of this synthetic sex pheromone, attracting a great many males, which then try to have sex with the dispenser, and as such fail to produce offspring, and the females cannot lay their eggs inside the developing berries. There's also increasing interest in using similar chemicals to attract predatory insects to vineyards, as well as creating refined tools to monitor insect populations overall. and within specific species. Physical control can also be done in small scales. For instance, a 5-meter-high screen barrier surrounding the vineyards can exclude sharpshooter insects, preventing the bacteria from spreading. Optimal amounts of water within the vines also act as a limiting factor within leafhopper populations. There's also a great deal of interest in new types of chemical compounds, specifically those produced by biological organisms. The current most popular biopesticide within vineyards is Bacillus thuringiensis, the bacteria species from which the Bt genes are extracted from to create Bt corn, soybean, and wheat. The bacteria infect Lepidopteran larvae, which include caterpillars from moths and butterflies. Interest in other biopesticides include different species of fungi, nematodes, and viruses. Biopesticides are not very often used, and there's very few available because these biological pesticides are living organisms and are unstable, i.e. die within heavy amounts of heat, UV radiation, or drying out. They're slow to act in comparison to conventional pesticides as well, and have a shorter shelf life due to being living organisms, but it's still well suited for organic production and other low input systems. And that about covers everything. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.